Hi everybody, today we're going to be talking about photosynthesis. When we talk about autotrophs versus heterotrophs, keep in mind auto means self, troph means feeding. So autotrophs literally can take care of themselves. They don't need any other organisms. Autotrophs can produce organic compounds from inorganic compounds. For example, they can take CO2 gas and convert it into sugar molecules, storing energy from the sun in the molecules of the sugar. Examples of autotrophs would be plants or algae. Heterotrophs cannot do this. Hetero means other. So other feeding means that heterotrophs have to consume organic molecules from other organisms and then break down those molecules for energy. Here's the overall reaction for photosynthesis. Autotrophs take energy from the sun, combine it with carbon dioxide gas and water, and are able to take that light energy and trap it and store it as potential energy in the covalent bonds of the sugar molecule. During this process, oxygen gas is produced as a waste product. The carbon is going to be gaining electrons and becomes reduced, forming glucose. The water is going to lose its electrons. It becomes oxidized to become molecular oxygen. Glucose stores the photon energy from the sun as potential energy, ready to be broken down and converted to ATP when the organism requires it. Photosynthesis happens in the chloroplasts. This is an organelle found primarily in the leaves of plants, but also in algae and cyanobacteria. The chloroplast is surrounded by a double membrane. Inside the chloroplast is a thick fluid called the stroma. Tiny pores in the membrane, called stomata, allow carbon dioxide to enter and oxygen to leave. A third membrane surrounds the flattened sacs called the thylakoids, shown here in green. The thylakoid membranes are green because they are full of a green pigment called chlorophyll. The chlorophyll pigment is what absorbs light energy and allows the plant to convert energy from sunlight to chemical energy stored in glucose. Pause your video to review the parts of the chloroplast. Photosynthesis is made up of two different processes. The first is the light reactions. These require the light from the sun, as well as water. Water is split into hydrogen and oxygen gas. The hydrogen ions are used to create a gradient, and the oxygen gas is a waste product that will leave the chloroplast through the stomata. The other product of the light reaction is stored energy in the form of ATP and NADPH, an electron carrier. The light reactions take place within the thylakoid membranes. The other process is called the Calvin cycle. And this is where the energy produced during the light reactions is used to bind together molecules of carbon dioxide to make sugar. This step does not require sunlight to occur. Plants can do this in the absence of light, so these reactions are sometimes referred to as dark reactions. The chlorophyll within the thylakoid membrane is a pigment. Its job is to absorb photon energy, which causes an electron within the pigment molecule to be kicked up to a higher energy state. When the energy is released, the electron returns to its original ground state. The pigment molecule can either release this energy as heat, as light, or it can transfer the energy to another chlorophyll molecule. We don't want to waste this higher energy state. We don't want to let the energy go, so we end up transferring that energy from one chlorophyll molecule to another within the thylakoid membrane. Excited. Lucky for the plant, their chlorophyll molecules are clustered together within a series of light harvesting complexes within the thylakoid membrane. These are called photosystems. The excited electron in the chlorophyll molecule is passed around from molecule to molecule until it reaches the special pair. This is a pair of chlorophyll molecules within the reaction center complex. These molecules are reduced by the electron transferred to them and will in turn pass along the high energy electron to the primary electron acceptor. Let's take a closer look at how this happens next. Within Photosystem II, Light is absorbed by the chlorophyll pigments. The light causes the excitation of an electron, which can be passed from chlorophyll to chlorophyll molecule until it reaches the reaction center complex. Special pair of chlorophyll pigments P680 receive the energy, which causes an electron to jump into a higher energy shell, and that electron gets transferred to the primary electron acceptor. The special chlorophyll molecules, P680, have been oxidized. They gave an electron away. In order to do this reaction ever again, we must now reduce the special pair. This is where water comes in. If we split a water molecule, the electrons from breaking the bond between oxygen and hydrogen reduce the P680 chlorophyll molecule. The hydrogen atoms are released into the thylakoid space, and the oxygen left forms oxygen gas, molecular oxygen, by pairing up with another oxygen from another reaction center. The electrons are passed on from Photosystem II to Photosystem I via an electron transport chain. As in the electron transport chain in mitochondria, the cytochrome complex is reduced by the electron, then oxidized as the electron moves down the chain. A little bit of energy can be harvested to pump a proton across the thylakoid membrane into the thylakoid space. This establishes a hydrogen ion gradient across the thylakoid membrane, which can be used by ATP synthase to create ATP. We've been talking about Photosystem II this whole time, and you may have wondered why it's called Photosystem II. Where's Photosystem I? Here it is. Some of the energy remains in the electrons after the electron transfer chain. The electrons are ultimately transferred to the special pair in Photosystem I. Photosystem I this whole time has also been gathering light energy with its chlorophyll molecules and transferring the energy to its own special pair, P700, which excites an electron to jump to a higher energy state and is given to a primary electron acceptor. But in this case, we don't use water to reduce the special pair. We use the leftover electron from Photosystem II to reduce the special pair, resetting it to be used again as more light energy is converted. The primary acceptor of the electron for Photosystem I also donates the electron to an electron transport chain, which uses the electron to reduce an electron carrier, NADP, to NADPH. It uses an extra hydrogen from the stroma, increasing the gradient between the stroma and inside thylakoid space. Hydrogen is actively being pumped into the thylakoid space by the electron transport chain of Photosystem II. It's also being used up by NADP reductase in reducing NADP to NADPH. This creates a large gradient. The large gradient forces ATP synthase to make ATP. What happens when you take away the light? This entire reaction will stop. The light reactions shown here convert light energy to potential energy. We use the products of the light reactions, ATP from Photosystem II and NADPH from Photosystem I, to run the Calvin cycle. The Calvin cycle, or the dark reactions, is the synthesis part of photosynthesis. This takes place in the stroma of the chloroplast. The Calvin cycle takes NADPH and ATP from the light reactions and uses these potential forms of energy to undergo carbon fixation. In other words, they use the energy to make sugar from CO2. There are three phases to the Calvin cycle: carbon fixation, reduction, and regeneration. The first phase is carbon fixation. Carbon dioxide from the atmosphere enters the stroma. 3 molecules of it enter the Calvin cycle at one time. These 3 molecules of CO2 each bind to a molecule of ribulose bisphosphate, or RUBP, a 5-carbon molecule. We started with 15 carbons and we added 3 CO2 molecules, which gives us 18 carbons. The reaction of CO2 and RUBP is catalyzed by an enzyme called rubisco. This is the most abundant enzyme on the planet. You now have 3 6-carbon molecules, or 18 carbons. Very quickly, each of these 3 6-carbon molecules are broken in half. Each one becomes a 3-carbon molecule called 3-phosphoglycerate. You still have a total of 18 carbons. The next phase is the reduction phase. During the reduction phase, the 6 3-carbon molecules are reduced to form 6 molecules of G3P, glyceraldehyde 3-phosphate. It requires 6 ATP and 6 NADPH to do this. These energy molecules are products from the light reactions of photosynthesis. We still have 18 carbons though. We only need 15 carbons to reset our cycle for another 3 carbon dioxide molecules to bind with RUBP. So we can remove 1 molecule of G3P as our output. Think back to glycolysis. Do you remember G3P? This is the molecule that we got during glycolysis when we first split our glucose molecule in half. It's a 3-carbon sugar. Put 2 of them together, you make glucose. 1 G3P leaves the cycle each turn. The remaining 5 G3P stay and are regenerated to get ready for the next turn of the cycle. This is the regeneration phase. It takes an additional 3 ATP to do this. For the net production of 1 G3P sugar molecule, it requires 3 CO2, 9 ATP, and 6 NADPH. So to review, during the light reactions of photosynthesis, these reactions are carried out in and across the thylakoid membranes. The photosystems of the light reactions convert light energy to chemical energy of ATP and NADPH. We're also going to be splitting water using the hydrogen ions to create a gradient and releasing oxygen gas. The products of the light reaction, NADPH and ATP, are going to go to the Calvin cycle to run those processes. Oxygen gas is released as a waste product. The Calvin cycle reactions take place in the stroma of the chloroplasts. We're going to be using ATP and NADPH to convert carbon dioxide to the sugar G3P. We're going to return ADP, inorganic phosphate, and NADP to the light reactions to do the cycle over again. That's it for today. See you in class.